Here's where we left it last time at mechanisms of hormone action, and that when hormones dock on target cells, first of all, the target cells have to have receptors for that particular hormone, and this is what's going to happen in general, that the target cells, it's going to change the membrane permeability, or those cells are going to undergo mitosis or to secrete substances. Something's going to happen. And recall that hormones are either going to be made out of amino acids in general. Most hormones are made of amino acids, or they're going to be steroid based. So here's our second slide on mechanism of hormone action. Now, the hormones, when they dock on a cell, they're going to make that cell do one of those actions, either undergo mitosis, change the plasma membrane permeability. They're going to make those cells do one of these things, either through second messenger systems or through a process called direct gene activation. Now, if I'm a hormone, am I going to use this direct gene activation to make my target cell do its thing, or am I going to use the second messenger system? Well, it depends on if I am made of amino acids, or it depends on whether I'm made of steroids. If I'm a steroid or a lipid-soluble hormone, then I'm going to use this process called direct gene activation. If I am an amino acid-based hormone, then I'm going to use the process called the second messenger system to influence my target cell. There's one exception to this. Thyroid hormone, this is very strange, thyroid hormone falls under this category of direct gene activation. I believe it's because it's just so small. It's a very small. Thyroid hormone is actually made of the amino acid tyrosine. It's just one little amino acid, so it's very small. So it's actually an amino acid based hormone, but it uses this little system here. But all of our steroid based hormones use direct gene activation to influence their target cells, while all of our amino acid based hormones use second messenger systems. So the two types of second messenger systems that we'll study are called cyclic AMP messenger systems and the PIP calcium second messenger systems, or it's called PIP calcium signaling mechanisms. So we have two second messenger systems that we'll go over in detail, and then I'll teach you about direct gene activation. We do these first, even though they're second on the slide, and then we do direct gene activation. So first, let's take a look at this cyclic AMP second messenger system. This is what it looks like. It's a little complicated, but if I am a protein-based hormone or an amino acid-based hormone, I can actually go inside of a cell, and this is a list of different types of hormones that use this CAMP second messenger system to influence their target cells. So I know you don't know what these mean yet because you haven't really studied the chapter, but this is adrenocorticotropic hormone, follicle stimulating hormone, luteinizing hormone, glucagon. This is parathyroid hormone, thyroid stimulating hormone. That's what these stand for. And then there's calcitonin. So these are the hormones that use the CAMP second messenger system. Please do not waste your time memorizing this. Doesn't matter at all which hormones use this. What I want you to know is that it's the amino acid based hormones that have to use second messenger systems. And the reason that they have to use a second messenger system is they have to use a second messenger to influence the inside of the cell, because this hormone cannot get inside of the cell. So it has to use a second messenger to get its message inside the cell. Because this is a polar substance out here. This hormone is polar, so it can't get through this phospholipid bilayer. If you'll recall, our cell membrane is made up of a phospholipid bilayer, and this is a non-polar substance here. So polar substances can't cross through it. If this polar substance wants to get inside of the cell, it would have to go through a gate, and we don't have a big gate that's big enough for hormones to get in. So what these hormones do is they dock on the outside of the cell on receptors. So here's our hormone receptor. So these are going to be the parts of our second messenger system that we're going to go through right now. So our hormone docks on the receptor. It's the first part of our second messenger system. And when it does that, it's going to activate a G protein. And this G protein was inactive. And it was inactive, and it had this thing right here on it. This is like an ADP molecule. An ADP and ATP. ATP is our energy molecule, right? GDP and GTP. GTP is an energy molecule, and it's just like ATP, and the difference between it is it has, instead of adenine, it has guanine in it. And so this is an energy molecule here. So think of GDP just like ADP. You've always heard of ADP before, and you've always heard of ATP. So this is just like energy. So this G protein was really close to our receptor molecule. It was docked on the outside of our receptor cell, wherever this cell is in the body. Could be in your big toe for all we know. But this hormone comes and it docks on the cell. And when it docks on this receptor, this G protein, which was inactive because it had GDP attached to it, this GDP jumps off and a GTP molecule attaches to it. And this G protein is now activated, and it will go across the cell membrane and attach to a different molecule altogether. And this molecule is what we call an effector enzyme, and it has a certain name in this messenger system, the CAMP second messenger system. That name is adenylate cyclase. And what our adenylate cyclase molecule is going to do is it takes an ATP molecule from within the cell, and it takes two of the phosphate groups off of it, and it converts it into a cyclic AMP molecule. If you look back in chapter 1, you'll see that a picture of if you take two phosphate groups off of this guy, and that's when we get cyclic AMP. And so what cyclic AMP is, that's our actual second messenger. We couldn't get this hormone inside the cell to make something happen, so we needed a messenger. That's our cyclic AMP molecule. What cyclic AMP does is it activates these things inside the cell called protein kinases that were inactive. They're just hanging out inside the cell. And when cyclic AMP is present, it will activate them. And these active protein kinases make something happen inside the cell. It might make the cell undergo mitosis. It might make the cell start secreting something. It might open up ion channels to increase permeability of the cell. It does these things on this slide right here that we were talking about. It might cause the cell to make proteins or regulatory molecules. It may activate or deactivate enzymes. It does whatever that hormone wants this cell to do. But the hormone couldn't get inside the cell because it's a protein-based hormone. It's polar. So that's what our cyclic AMP second messenger system does. Now we had two second messenger systems. We're about to go over the second one, the PIP calcium second messenger system. It's just like this one, but we have different players. We're going to have a different effector enzyme and different second messengers. But one thing I need to tell you before we go to it, we have a huge cascade effect. Cascade effect means there's a lot of amplification. A little bit of hormone goes a very long way. One little hormone molecule can dock here, and we can have thousands of protein kinases that are activated. So that's what cascade effect means. Huge amplification. A little bit of a hormone goes a long way. Now because of that, we have to have an enzyme that's going to degrade this, degrade the CAMP molecule as fast as it's produced. When we produce this CAMP, we want to break it down really fast, so there's a molecule inside the cell called phosphodiesterase, and this phosphodiesterase is our enzyme that's going to break down that CAMP to make sure that this hormone is, the effect of the hormone is very short-lived. So let's go into our second messenger, the second messenger system, which is the PIP calcium second messenger system. Now you don't have this picture in your book anymore, so you might want to print this page off. Here are some examples of some hormones that use this second messenger system. And again, please don't take any time memorizing which hormones do this. The point you want to take away is if you see these hormones here, then you know that these hormones are amino acid based hormones. So we have a hormone that's going to dock on this receptor. And well, let's just ignore this first picture. The hormone docks on this receptor, and what happens is that here's our G protein, and it was inactive because it had a GDP that was attached to it, which is displaced by GTP, and the activated G protein will go along. And now here's our effector enzyme. This effector enzyme is now replaced. This effector enzyme is called phospholipase C. in this system. And what phospholipase C does is it breaks down this molecule. This PIP2 molecule is actually called phosphatidylinositol biphosphate. That's its real name. And it's going to break down into this molecule called DAG. DAG is really called diacylglycerol. And it breaks this PIP2, or phosphatidylinositol biphosphate, breaks into diacylglycerol, and it also breaks into this IP3, which is inositol triphosphate. So we break PIP into DAG and IP here, and this molecule right here is what's going to, this diacylglycerol is going to be our second messenger. So this second messenger activates our inactive proteins and our kinases, and then something happens within the cell. But this phosphatidylinositol biphosphate, when it breaks down into the inositol triphosphate, this will go into our endoplasmic reticulum, and if you'll recall, endoplasmic reticulum sequesters calcium. I hope you remember that from last semester. When it releases calcium inside the cell, calcium will also act as a second messenger. So that's why up here calcium is listed as a second messenger. So calcium will also trigger a response within the cell. I know you're so excited you can't stand it. This is our second messenger system. The big takeaway message here is that the hormone can't get inside the cell because it is amino acid based. So we need second messengers to do the dirty work, to activate protein kinases to make something happen within the cell, and calcium is one of those second messengers. So we had two second messenger systems. We had the PIP calcium second messenger system, and we had the CAMP second messenger system. And amino acid based hormones are the ones that use second messenger systems. Now if we're going to go back and review for a second, we had two mechanisms of hormone action. We had second messenger systems, and these were used by amino acid based hormones. So we just looked at these two types of second messenger systems. There are other second messenger systems. These are just the most popular ones that have been studied the most. Some amino acid-based hormones don't use second messenger systems. For instance, insulin does not use a second messenger system. You do not have to know the mechanism by which insulin gets into your cells. You're lucky for that. It's quite complicated. But I would like, if you have access to the website, for you to watch the little video to see how insulin gets into your cells, because it's quite fascinating. Now, on the direct gene activation. Direct gene activation, it is used by the steroid-based hormones. Now remember, steroid is a lipid. That's why we can say in lipids dissolve in lipids. That's why in this slide we say used by lipid-soluble hormones, because lipids dissolve in other lipids. So steroids can go right through the plasma membrane, and they can go right in to where your genes are. So that's why this name it comes about, direct gene activation. So let's take a look at that. And then we have this strange one, thyroid hormone. Direct gene activation is much more simple. So here we have a steroid hormone, and it can go right through our plasma membrane, and it doesn't have to worry about anything. And it can go right into where our DNA is. And our DNA right here, all the collection of all of our DNA is called chromatin. We have our receptors actually we have sites for our receptors actually on our DNA. Most of the time our receptors for the hormone aren't docked on our DNA. They're floating around out here. So let me take a step back. Here we have our receptor for our hormone. And our hormone's going to come into the cell, and here's this receptor. And see this little green thing on it? This is called a chaperone. So here's a chaperone. A molecular chaperone is a protein. Now, molecular chaperones are amazing little things. They do quite a bit of things, quite a bit of they do just as the name suggests. They chaperone things. And in this case, this molecular chaperone is chaperoning this receptor. If we just had this receptor floating around with your very fragile DNA, it could put little nicks and dents in it and damage your DNA. But if we have this molecular chaperone attached to the receptor, then it protects your DNA. So we have this receptor chaperoning complex when the two are attached, and they're floating around waiting for the hormone to come in. So when our steroid hormone comes into place and attaches to it, then our molecular chaperone detaches, and now we have our steroid receptor complex. So here it is, right here. It sort of looks like a, what is that, little candy corn? Yeah, kind of makes me hungry. Okay, so our receptor hormone complex is ready to dock onto our DNA. So it docks onto our DNA, and it turns on a gene. It makes something happen. Now your DNA can directly make an mRNA, a copy of the gene, an mRNA molecule. This is a copy of the DNA itself. This is a copy of the gene from the DNA molecule. When you make a copy of a DNA molecule of a gene on DNA, that's called transcription. And so this mRNA molecule can leave the nucleus and go right out here and feed through these little ribosomes. These are the little organelles that make proteins, and when they feed through and make proteins, that's called translation. And there we go. We make little proteins. Something happens. So steroid hormones, they go right in, they dock with the receptor binds with the DNA, and we make proteins from that. So the reason this is called direct gene activation is because these steroids activate genes on the DNA. So we have a list of questions here for you. What type of hormones use direct gene activation? I hope you said steroid hormones, but we also have another little flunky one. Remember that thyroid hormone does this. Thyroid hormone's a little strange. We don't have, the receptors are bound to the DNA already. What is transcription? Transcription is right here. When you make a messenger RNA molecule, a copy of the DNA, of a gene on DNA. What is translation? Translation is when you make a protein off of a ribosome, or the ribosome actually makes the protein. Which organelle is responsible for translation? Whoops, sort of told you that already. Ribosome. Why is the molecular chaperone needed? Yep, to protect the DNA. Molecular chaperones are really cool in other instances as well, because when this protein is being made, there are also molecular chaperones out here folding the protein, making sure they're nice and folded the exact right way. And there are also chaperones. There are all kinds of chaperones. There are heat shock chaperones. You should look all these up. Look up molecular chaperone. Look them up in Wikipedia and see all the jobs chaperones do. Okay, target cell specificity. Now, target cells have receptors. We've seen that. We saw that in this case, here's the receptor for steroid hormones. We have receptors here for our second messenger for these amino acid-based hormones. If this is oxytocin, if this hormone's oxytocin and oxytocin binds right here, and this is a uterine cell, then this cell will start to have contractions. And then, you know, you're just in for the ride of your life. God bless you. If this was oxytocin floating through the blood, and this was a toe cell, then there's not going to be a receptor on that toe cell for oxytocin. Can you imagine if you had some kind of some sort of mutation and you had receptors for oxytocin on your toe, and then you would have contractions and then toe contractions at the same time? That would be horrible. But anyway, you have to have a receptor for a hormone to have effect of that hormone. So let's see. So target cells have receptors. There's target cell specificity, and the reaction to that hormone is going to depend on how much hormone is in the blood. If you have very little hormone in the blood, you're not going to have a big effect of that hormone. Think of it this way: if you're a bodybuilder and you have a lot of testosterone, have you ever seen somebody with too much testosterone? Or think of a woman who we all have all females have just a little bit of hormone. It's produced by our adrenal glands. Have you ever seen a woman with too much testosterone? You know that woman because she has a beard and a mustache. So our reaction to our hormone depends on hormone levels, and also the amount of receptors our target cells have. If we lose our receptors on our target cells to our hormones, then our cells can't respond to hormones. Plain and simple. No matter how much hormone you have in your blood, if you lose your receptors to that hormone, then your target cells can't respond to them. For instance, if you have, for insulin, if you have as much insulin in your blood, but you don't have any receptors for that insulin, then your body cells can't respond to the insulin. So you need the receptors. And affinity for a hormone means some receptors really respond very well to a hormone, and they have a strong affinity for the hormone, and then some receptors don't. It's sort of like if you buy really cheap Velcro or very high quality Velcro. Okay, on the upregulation and downregulation. Sorry that I'm whipping through these so fast, but it's hard to read definition. The case of upregulation. Sometimes if you have high blood levels of a hormone in your body, then your cells will respond by making more receptors for that hormone. On the other hand, sometimes when you have very high blood levels of a hormone, your cells will respond in an opposite manner, and that's called downregulation. Sometimes if you have high blood levels, your cells will rebel and get rid of receptors. So these are two different ways your cells respond to high blood levels. Okay, on the half-life of a hormone. A half-life of a hormone is the amount of time it takes for your blood. Say you release 100 micrograms of a certain hormone. It's the amount of time it takes for you to only have 50 micrograms of that hormone in your blood. So that's what the half-life means. And onset is, does it take 1 second for that hormone to cause an effect, or does it take days? So onset can be immediate. You can have an immediate effect, or it might take days for the effect of the hormone to be seen. And usually to be days, that would be like your steroid hormones. Most all hormones are pretty fast except for the steroids. Now, how long a hormone affects you, the duration, could be seconds to hours. And you think, surely hormones last longer than that, but you have to produce hormones at a continuous rate for them to affect you. Now, going back to this, when hormones circulate in your blood, they're going to circulate either free, that's by themselves, or bound to a substance like a carrier molecule. The hormones that are bound to carrier molecules are your steroid molecules and thyroid hormone. They're the ones that need carriers. So the lipid-based hormones need carriers and thyroid hormone. So the amino acid ones are free. When it comes to hormone interactions, that's how hormones are going to interact with one another. We have 3 different definitions for you to know: permissiveness, synergism, and antagonism. Permissive hormones are when one hormone needs another to be present to work, and this is actually very common. And the best example would be thyroid hormone, because it works with so many other hormones. TH stands for thyroid hormone, and growth hormone. Thyroid hormone, when you're growing, if you don't have thyroid hormone and growth hormone together, you're going to be a little person. Basically, thyroid hormone affects all your other hormones. So take thyroid hormone away and you're going to be a mess. Synergism is when 2 hormones produce an amplified effect. And the example here would be all your hyperglycemic hormones, all of the hyperglycemic hormones. What hyperglycemic means is elevated blood sugar. Emia means blood, hyper means more than, and glyc means sugar. So these are the hormones that would cause an increase in blood sugar. And so, for instance, glucagon. Glucagon, what hormone? What? Let's say this. Which organ releases glucagon? Do you remember? It's the pancreas. And when the pancreas releases glucagon, it targets the liver, and the liver will, what will it break down? Do you remember? It will break down glycogen to release glucose, and that will increase your blood sugar levels. You remember glucagon? You might have to look back to your chapter 1 notes, but that increases blood sugar levels. Well, usually when that's happening, there's another hormone that's being released, epinephrine. Epinephrine will do the same exact things, and so that you'll get an amplified effect. And we'll go on, we'll talk about a lot of hyperglycemic hormones. We have quite a few in this chapter. Antagonism is when two hormones work against each other. For instance, if you have a hyperglycemic hormone versus a hypoglycemic hormone. We've just listed two hyperglycemic hormones, and you know of a hypoglycemic hormone. A hypoglycemic hormone is a hormone that reduces blood sugar levels. What hypoglycemic hormone do you know of? Insulin, right? Insulin. Insulin reduces your blood sugar levels because which organ releases insulin? Right? When the pancreas releases insulin, it tells your body cells to take up blood sugar. And when it tells your body cells to take up blood sugar, that reduces your blood sugar levels because the sugar goes from the blood into your body cells. Also, it tells the liver to take the sugar inside of it, and it tells the liver to take that glucose and make it into glycogen. And when it stores it as glycogen, then later on, the liver has something to break down. Another example of let's see, do I have another example of antagonism for you? I do. Say progesterone causes the loss of estrogen receptors on the uterus. So that's not good because it has an antagonistic effect on estrogen. Estrogen has to hate that. So that's another example of antagonism. Oh, thank goodness. I was wondering if this lecture was ever going to be over. Okay, that's it.